Hello viewers, welcome to my channel. In today's video, I will be discussing about Jak STAT Pathway. This is also known as Janus Kinase Pathway or Janus Kinase STAT Pathway. This is very much alike with that of tyrosine kinase pathway or tyrosine kinase receptor mediated action. The last video which I had made is all about the tyrosine kinase. So similar to tyrosine kinase, when this Jak STAT pathway or Jak STAT receptor, they are not in bound form with the ligand, then they exist as monomer. So now coming to the ligand of this type of receptor, there are heterogeneous type of ligands which acts through this type of receptor or this pathway, and they can be the cytokines. The examples are like interferon gamma, then growth hormone, prolactin, and erythropoietin. These all act through this type of Jak STAT pathway. So when it is not in bound form, then it exits as monomer. In monomer, we have the extracellular part, the part which is linked with the membrane, and then intracellular part. The difference which is there in comparison to tyrosine kinase is that we don't have the intracellular enzyme in this Jak STAT pathway or this type of Jak STAT receptors. Whereas tyrosine enzyme or tyrosine kinase used to be there in the intracellular part. So you can say that this particular receptor has no intrinsic enzymatic activity. Rather, it will cause the binding of Jak separately. We'll see it in details later on. So I will explain the whole pathway by the help of steps and then figure. So what happens initially, as we know that it exists as a monomer, the receptor. So ligand would be there. Ligand can be any one of these. So ligand will bind with the receptor. Okay, ligand will bind with the receptor. So it will be the same as that of tyrosine kinase. In that tyrosine kinase or Jak STAT pathway or this type of receptor, we have 4 subunits, 1, 2, 3, 4. The details of which I have already told in the previous video. So ligand will bind with the receptor and they will have a specific site where the ligand can bind. So here the example which I have taken is cytokines. So the receptor is having extracellular part, membranous part, and then intracellular part, but this intracellular part has no enzymes attached with it. So what happens later on, we'll see. The ligand binds with the receptor. So here two molecule of cytokines goes and binds. So one molecule will bind over here in one monomer, another molecule will go and bind to another monomer, and then dimerization occurs. As you know, dimerization occurs by this 1 and 4th subunit. Sorry, 2nd and 4th subunit. So dimerization occurs, and then dimerization will recreate Janus kinase in the intracellular part. So Janus kinase will be existing separately inside. Upon binding of ligand with the receptor, there will be dimerization occur, and this dimerization will recreate this Janus kinase from inside, which exists in separately, to the intracellular part. So this Janus kinase gets attached with the intracellular part, and then phosphorylation or activation of this Janus kinase occurs. Phosphorylation will make this Janus kinase active. Now what happens later on? So this recruits Janus kinase to cytoplasmic tail. So Janus kinase is being recruited. Then transphosphorylation of Jak over. Then what happens? This phosphorylation will in turn again cause the recruitment of another protein which is called STAT. STAT. So this phosphorylation of Janus kinase will recruit another protein called as STAT. That is signal transducer and activator of transcription. That is the full form of STAT. You will find the full form in the description box. So this comes and binds to the Janus kinase, and then it gets phosphorylated by Janus kinase. So STAT gets phosphorylated by Janus kinase. So recruit STATs and then phosphorylation occurs. After that what happens? Then there will be dimerization of the STAT part. This STAT dimerization occurs, and this dimerization occurs by the phosphates of another STAT. So there is one STAT. This will dimer by the help of phosphate of tyrosine of this part, and then this will be causing the attachment of forming a dimer with this part by the help of phosphate of tyrosine of this STAT. In that way, there is formation of STAT dimer. The STAT dimer formation occurs, and then that will be dissociates, and the formation of dimer occurs, and then it translocates to nucleus. It goes to the nucleus. So clear? We'll see again here. There will be formation of dimer, and then it translocate to the nucleus, and then nucleus there would be regulation of transcription, which will cause the formation of the specific protein, and then the action would be given. So you might face difficulty in making this type of receptor. So you need not make this type of receptor. Just to explain, I have told the extracellular part as 1, 2, 3, 4. You can simply make a receptor like this membranous part we have, and then the intracellular part we have, and then here some sort of like this receptor you can make. This type of receptor will do, or any type of receptor you can make at extracellular part. No need to make on all the subunits. That was for understanding sake. So I'll just briefly explain it. Ligand binds with the receptor, then receptor dimerization occurs. Here is happening. Then it recruits Jak to the cytoplasmic tail. So recruitment of Jak here to the cytoplasmic tail. Then there is phosphorylation of Janus kinase appears. Then it recruits another protein here, and then that gets binds with the Jak. So recruit STAT, and that phosphorylation of that also happens. Then there is dimerization of STAT occurs, that dissociates from this Jak and then goes to the nucleus, and then from nucleus there is formation of transcription factor proteins, and then ultimately the effect would be given. So this was all about Janus kinase STAT pathway. Thank you so much.